Oh, good. We're approaching almost 10 years working on this project, um, and it started when I was a PhD student at Ohio State University. But everybody, unfortunately, that we've run into that's growing vegetables in high tunnels will run into soil-borne disease issues at some time. So consider yourself forewarned and forearmed. And we'll talk about an important tool today to manage these soil-borne diseases. Okay, so let me get my screen. Okay, so just a quick orientation in case you're not familiar. You guys all know where you are at in Nova Scotia, but I'm down here in Ohio. We have actually a very similar climate. When Talia and I met initially, the timing for planting in high tunnels is very similar. So our growers in Ohio will plant March, April, and then they will harvest September through November for whatever crop they're growing. The majority of our growers will grow tomatoes. That's our major cash crop. But we also have some growers that are growing cucumbers. There's a few leafy greens growers in high tunnels. And I think some of our growers are also switching into melons. But again, tomato is the dominant crop. And unfortunately, we're seeing a lot of high tunnels that are starting to look like this. Now, if I showed you this picture and asked you, when do you think this picture was taken? You might say August or September. These are tomatoes that are near the end of their season. But this picture, unfortunately, was taken in June. And these are tomatoes that are very stressed by soil-borne diseases. You can tell that this crop is not thriving. And what I'm seeing as a plant pathologist when I look at this crop, I see that these plants are stunted. Normally, this variety of tomatoes, which is red deuce, should be at the top of these stakes. There's a lot of dieback. The plants are wilting and they're yellowing. And what that indicates to me is that there's an issue in the soil, and it's likely that we have a disease issue in the soil. So when we're thinking about looking for disease issues in our soil, it's easy to see foliar diseases, but when we have to look for soil-borne diseases, you have to dig a bit deeper because of the hidden nature of roots. So what you as a grower can look for on diseased roots, crown, and stems that would indicate if you have a soil-borne disease problem, there are some key things to look for. So you can look for girdling, that means your stem is pinched, or browning at the soil line. You can look for internal stem discoloration. So just cut that plant at the base, look for some browning within the stem. Your roots can be reduced in size, and oftentimes you'll lack fine roots because a lot of these soil-borne pathogens like to nibble at the tiny roots of your plants. Roots are often discolored, either golden, brown, or black. And on this picture right here, you can see this is a very unhappy root system. And a healthy tomato root system should be white, but these tomato roots are just very dark in color. Depending on the pathogen, sometimes you can have soft roots that fall apart easily. This is a good indication that you have an oomycete pathogen like Pythium or Phytophthora. Roots can also change in texture and they can become very barky. And there's one specific disease that we'll talk about. And then also you can have galling and club-like growth on roots. This usually indicates that you have nematodes. Now, this is one of the worst root systems I've seen in a high tunnel, and this has root knot nematodes, quirky root rot, black dot root rot, and verticillium wilt. So this poor plant is hit by a lot of different diseases. So, as I said, roots can be hit by a lot of different diseases. And what we're seeing in our high tunnels is that these soil-borne diseases can occur alone, but more often what we're seeing is that these diseases occur in complexes, which means that you have one or more diseases or two or more diseases occurring at once. So what we're seeing is we have our fungal wilts, verticillium and fusarium wilt. We have our fungal root rots, quirky root rot and black dot root rot. We have damping off caused by oomycetes, either Pythium or Phytophthora, or fungi, Rhizoctonia, or other fusarium species. We have white mold caused by Sclerotinia sclerotiorum. And then for nematodes, root knot nematodes are the dominant nematode that we're dealing with. Now, I just want to walk you through some symptoms to keep an eye out for if you're growing high tunnel tomatoes. And a lot of these symptoms and other vegetable crops can be similar. So if you are growing a different crop, just kind of tuck these away in your brain. as things to look out for. So verticillium wilt is caused by the fungus Verticillium dahliae, and the distinctive symptoms that you're going to look for this disease are V-shaped lesions on the leaves. So you're going to see a dead area of the leaf, it's V-shaped, and that's going to have some yellow discoloration along the margin. For both verticillium and fusarium wilts, what you want to do if you suspect this is sacrifice a plant or two and cut into those stems. And you want to look for discoloration, this browning, right under the outside of the stem. And what that indicates is that the vascular tissue of your plant, what conducts nutrients and water, is actually infected by the fungus, and that's blocking the flow of those water and nutrients. Now, a good indication that you have verticillium wilt and not fusarium wilt is that verticillium wilt has a very wide host range, and this usually leads to problems in other crops. So this is a grower that I worked with in Southern Ohio, and he was rotating pepper and pepper and strawberry in his high tunnel, and he was having verticillium issues in both crops. You can see that these strawberries have a lot of wilting and dieback, and those are very distinctive symptoms of verticillium in strawberry. Now, fusarium wilt, which is caused by Fusarium oxysporum formospecialis lycopersicii, don't worry too much about the Latin names. I just put them up so that if you are trying to compare diseases, you can make sure you're looking at the same disease. But the key symptom here is that usually the yellowing and dieback associated with fusarium wilt only occurs on half the plant at once. It's a very unusual symptom, and it's just based on how the fungus actually infects the stem. So this is actually a picture of a tomato vine that's about 6 feet long, and you can see that that wilting and dieback is only on half of the stem. All right, now the next disease that I want to talk about is quirky root rot, and this actually is probably the most important for us in more temperate regions. Unfortunately, this is a disease that we didn't identify until 2015 in Ohio, and it's a lot more common across the Midwest and Northeastern United States. It has been identified in Canada and Ontario, so it is an issue in Canada. So just be aware of this disease. But quirky root rot is caused by the fungus Pyronicata lycopersicii, and this is very distinctive. It makes the roots look very barky in appearance and cracked. So, at the end of the season, make sure you're checking your roots. Make sure you're checking for this cracking browniness, this cracking brown change in texture of your roots. Now, if you have very early stages of quirky root rot, you'll see this cracking and browning in characteristic bands. So you'll have a healthy root, cracked brown root, and then a healthy root. Now if you see this type of symptom, this means that you usually have less pyronicata in your soil, which is actually a good thing, because you can start doing some management earlier on. We also have black dot root rot, which is caused by Coleoptera cocodes. This is the same fungus that causes anthracnose on tomato fruits. If you've grown tomatoes, I can guarantee you've probably had anthracnose on your fruits. Even if you eat tomatoes, I'm sure you've seen anthracnose on your fruits. It's these black sunken lesions on the fruits. But on the roots of the plant, these infected roots are golden brown and speckled in appearance. And what that speckling is you're actually seeing the fungus. And then if you look closer, sometimes you need a hand lens or a magnifying glass to see those speckling on the roots. So if you have that, make sure you're checking your roots a little bit closer. Okay, and then we have root knot nematodes. These are very easy to diagnose if you have them. What you want to look for on your roots is you want to look for a bead-like galling. The worse the infection, the larger and more misshapen these galls will get. If you do have root knot nematodes, I would suggest reaching out to your extension services and making sure that you address this issue as quickly as possible. Okay, and then we have some more minor diseases. We have rhizoctonia. The key symptoms you want to look for here are girdling and a dry brown root rot. It's very dark, it's dry, it's hard. And you can compare that to the type of root rot you get with Pythium and Phytophthora, which are oomycetes. That's a soft and wet root rot. And it's usually gold to brown in coloration. So, knowing your plants and what they should look like when they're healthy and when they're sick, you can kind of have an idea of what you might be dealing with depending on how these different root rots look. And then the last disease is timber rot or white mold caused by Sclerotinia sclerotiorum. This is a disease that a lot of our tomato and vegetable growers run into because this pathogen has a very wide host range. But what you want to look for is you want to look for a bleaching on the stem. That bleached area is going to be hollow, and if you open up that area, you're going to find black structures that kind of look like mouse turds or rat turds. And that's the best way to describe them. That's the survival structure of the fungus. And if you see this in your high tunnel, make sure you get those plants out as soon as possible because you don't want those survival structures to stick around for too long. Okay, so that was a really quick overview of what you want to look for. And I do have a resource sheet that I gave to Talia that she'll be sending out to you. And I can also send a PDF to Talia to send out of the slides for your future reference. But so you know what to look for, but I want to prepare you with some potential tools. So when we think about managing soil-borne diseases, there's two things we want to do. We want to prevent existing pathogens in our soil from increasing. And so to do this, we need to keep the disease pressure low. So keep those pathogens from reproducing in our soils. And to do this, we want to use management practices that promote plant and soil health. So we want to add good organic matter to our high tunnels. We want to use balanced fertility. And when possible, we want to reduce soil salinity because the soil salinity seems to be an issue that everybody's running into high tunnel use. And that can add extra stress to plants that can increase these diseases. And then you also want to use management practices that prevent pathogens from multiplying. So this includes the use of resistant varieties, grafting onto disease-resistant rootstocks, or rotation. But unfortunately, in a lot of high tunnels, this is valuable land that you have and you're growing the same crop year after year. So the next thing we have to do is the second practice for managing diseases is actively reducing existing pathogen levels. And this usually involves some form of soil infestation. Now, Talia asked me to talk about what current management options are, and I'm most familiar with rules and what growers do in the United States. But one tool that our growers don't have in the United States is use of most chemical products, such as fumigants. And open field fumigants can be used commonly for soil-borne diseases, but there's only one labeled for hoop houses in the United States. Now, I know your chemical labeling in Canada is slightly different, but still, I would not suggest using a fumigant in a high tunnel because you don't want to kill off the biology of your soil. There's also some fungicide drenches that are effective for oomycetes and rhizoctonia. If you're having issues with that, you could use those in your high tunnel. Those are options for you. And biologicals are really the other option that our growers are using. So bacillus or trichoderma products, some of them work, some of them don't. It all depends on your local context in your high tunnel. Now, some of our growers are using grafting onto disease-resistant rootstocks. For all the diseases that I talked about today, grafting is really effective for verticillium wilts. It can work for some of our fusarium wilts, and it also is very effective for corky root rots. Unfortunately, if you have root knot nematodes, grafting doesn't work as well for root knot nematodes, but it can provide you sometimes with a more vigorous root system, and your plants can kind of outgrow pathogen problems. So you can kind of get a little bit ahead of the problem. In northeastern United States and starting in the Midwest, our growers are re-looking at steam sterilization. So getting in a portable steamer and steaming their soils to reduce these soil-borne pathogens. Now, specifically looking at root knot nematode, there are some newer bionematicides that have pretty good efficacy against root knot. Now, these need to be combined with different management practices such as rotation to make sure you're keeping those root knot nematode populations down. And these are biologicals, so they're derived from microbes, and two examples are magistine or melicon. Now, unfortunately, I believe magistine is not currently registered in Canada, but I believe melicon is. Now, what that leads us to is the practice of anaerobic soil disinfestation. And this is a practice that we first started studying in 2015, like I said. We've had growers adopt it and use it successfully in Ohio, and in 10 other United States in the United States. So anaerobic soil disinfestation, or ASD, is a really complicated name for a disease management strategy, and I've never come up with a better name. But anaerobic just means without oxygen or air. So we're trying to get rid of extra oxygen in the soil, and that's going to lead to changes that cause soil-borne pathogens to reduce. So what we do to get rid of this oxygen is we first amend our soil with a carbon source, like wheat bran or midge or molasses. Then we irrigate that soil to saturation. And what we're doing here is we're filling all the air-filled pores in the soil so that we can reduce places in the soil where oxygen would reside. And then we cover that soil with a plastic mulch for 4 to 6 weeks. Now, what happens in this process is that beneficial microbes in the soil that are already living in your soil and are there start to break down that added carbon source. They get this huge source of food, and then they break it down. They start using up all the oxygen in the soil. So the soil goes anaerobic. As the soil goes anaerobic, that changes the chemistry of the soil, and you have reducing conditions that occur. These microbes make a lot of weird metabolites when they're breaking down things anaerobically. So they produce gases like carbon dioxide and methane. They also produce organic acids like lactic and acetic acids. And then they produce antimicrobial volatile organic compounds, or VOCs. So all of these different conditions that happen in ASD kill off soil-borne pathogens. Now, in temperate regions, we can successfully use anaerobic soil disinfestation. Now, other disease management strategies like soil solarization don't really work very well in Ohio, and I'm assuming they also probably don't work very well in Nova Scotia, because those rely on having solar heating to heat your soil from the sunlight, and it just doesn't get warm enough here to do that. But ASD is less dependent on temperature than these other practices. And luckily, this practice is effective against all of the pathogens that we have been finding in high tunnels. So it's very effective against root knot nematodes. It's effective against verticillium wilt, corky root rot, and black dot root rot. Now, a caveat is that fusarium, wilts, and root rots, that seems to be what ASD is least effective against. So if you're dealing with those, we might want to look at some more integrated management approaches. Now, what our growers are doing in Ohio that fits in best with their growing schedule is that in the Midwest, most of the time our market is glutted with tomatoes in August and September. So some of our growers will decide to pull their crop early and treat their soils with anaerobic soil disinfestation in September or early October. And the temperatures are still warm enough that if they close up their high tunnel, they can get good treatment efficacy. So usually we need about 4 weeks of tarping. If soil temperatures are cooler, you can just leave that tarping or plastic mulch layer on longer, and you'll get good treatment. Now, again, it's important whenever we're using one key disease management practice that we are making sure that we're combining this with other disease management practices, because we don't want to rely too heavily on one thing, because we might be missing a specific pathogen. And we also want more options to make sure that we have multiple tools that we're using to reduce these soil-borne pathogens in our soil. Okay, so let's step through step by step what ASD actually looks like, just so you have an idea of whether or not this might be practical for you to do in the future. Again, on that resource sheet, there is a link to a virtual workshop that I did with a grower collaborator and another collaborating university in the United States that steps you through step by step the whole process of ASD. So the very first thing when we are starting the ASD process is choosing a carbon source. And this is a food source for our microbes. So what we want is we want something that's inexpensive, and we want something that's locally available. So what we have available to us in Ohio might differ for you from Nova Scotia, but don't worry, there's a lot of good options you can use. What we look for most of our resources for carbon sources is we're going to go to a feed mill and see, what are we feeding our animals locally? It's inexpensive, it's easy to get, and we'll pick one of those. So what we found works best for us here is either wheat midds or wheat bran, soybean meal, molasses, or distillers dried grains. Now, these are applied at a rate of 6 to 9 tons per acre. So it is a pretty high amendment rate, but within a high tunnel, the cost of buying the carbon source, at least for us in the US, is around $250 US. So, if you're thinking about doing this treatment every 3 to 5 years, it's not really beyond the scope of reason that it's too expensive. Now, the best combination that we have found is the use of either wheat bran or wheat midds at 9 tons per acre, and then molasses at 4.5 tons per acre. And that wheat bran or midds is your core food source. And how the molasses acts here is the molasses is like a microbial lighter fluid. It's very sugary. The microbes really like it. They start to break that down really fast, and then they move on to the wheat bran or midds, and then there's plenty of food for them to make that soil go anaerobic. So what you do first in ASC after you have your carbon source is you spread it in your high tunnel. So this can either be by hand on small scale. So these are pictures of small scale and large scale trials that we did with different growers. Now, if you're treating your whole high tunnel, you also have the option to treat flats and treat all the soil, or you can treat just the area that you're going to use to make beds. So that's up to you. You're just gonna have to make slight differences depending on what you choose, but both will reduce diseases in your high tunnel. So in the small scale, you can just spread it by hand or in buckets. On the large scale, use a hopper spreader, and that'll help speed up the process. Next, we have to incorporate the carbon source into the soil, and we want to work that to a depth of 6 to 8 inches. And we just do this with a walk behind rototiller or a small pole behind rototiller that fits in your tunnel. Now, this depth of 6 to 8 inches, this is the area of the soil where we have the most microbial activity. If you can incorporate deeper, great. You're probably not getting as much as an impact at those lower depths, but you really want to make sure you're getting at that 6 to 8 inches deep. And then if you are treating just your beds, this is the stage that you would form your beds for planting. And the nice thing is you can treat your beds with ASD, and then the following season, just plant right into those beds. You don't have to remake them again. Now, if you are using molasses on a small scale, you can use watering cans or a backpack sprayer that you removed the spray nozzle from. On the large scale, you can use a sprayer. Molasses, if you've worked with it before, is really difficult to work with. It's very sticky. It clogs everything. So make sure you're diluting that either 1 to 2 or 1 to 4, because otherwise you're going to run into an excessive amount of clogging. All right, for irrigation, this is the second step of ASD. We want to make sure that we are saturating those soils, and you want to saturate to the point where water begins to pond on top. We don't want to flood your high tunnel, but you want to make sure that really you can't get much more water in. So that water needs to penetrate to the depth that you've worked your carbon source. So make sure that that water is saturated 6 to 8 inches. If you have a very sandy or well-drained soil, you might need supplemental water to keep that soil moist throughout the treatment. But it's been pretty rare that I've run into high tunnels where this is an actual issue. But most growers know their soil and how long they have to irrigate to get that soil nice and saturated. All right, then we want to tarp with plastic. On the small scale, if you're treating in beds, just use plastic mulch to cover your beds. And you want to make sure that this is a decent heaviness of plastic mulch. You can also use construction plastic or silage tarping. We also have growers that will use the old plastic coverings of their high tunnel. That works great and doesn't provide any extra cost. If you do have holes in any of your plastic, just make sure that you cover those up with duct tape because you want to prevent any air exchange. Now, this is probably the slowest step of ASD because when you are laying this plastic, you need to make sure that you cover or seal the edges of your plastic with soil or boards or bricks. And you're doing this to prevent air exchange. Again, we want to make that soil go anaerobic And if we have wind coming into our high tunnel and it's getting under that plastic, that's going to reduce the efficacy of our treatment. Now, our soil temperatures, I'm sorry for the Fahrenheit system here, should be above 68 Fahrenheit for at least the first 2 weeks. Now, the warmer you keep those soils and the longer you keep those soil warm, the better treatment impact you will have. So again, like I said, very early on, is this treatment lasts 4 to 6 weeks. Usually if you have temperatures above 68 Fahrenheit, 4 weeks is plenty. But if you're getting into an October, a lot of times your nighttime temperatures are pretty cool. So you might want to just keep those soils covered into November. All right. Now, after we do anaerobic soil disinfestation, the microbes in our soil have changed dramatically. So this is a picture of a soil after it's been treated with ASD. And there's some things that we can look at the soil that will indicate that we have a good treatment. The first thing you'll probably notice is that this soil smells pretty bad. If you've ever smelled a pond or a lake that's gone anaerobic, you know that smells very nasty, kind of like the backside of a cow. And that's a smell you're looking for to indicate you had a good treatment. You also want to look for this darker coloration. Those are metal ions in your soil that have become reduced. That's a really good indication that treatment has gone well. And don't be surprised or alarmed if you see a fungus or growing on top of your soil. These can be white, brown, yellow. This just indicates that you had a lot of microbes growing successfully. And those ones that are growing on top of your soil, those are just some ones that are taking advantage of any residual food that you put in your soil. So these are all good things to see following ASD. Now, for planting following treatment, you can either remove that plastic completely, or if you treat it in beds, just cut your planting holes. And you want to allow that soil to drain or breathe for about 1 week. I said there's this nasty smell. And there's just some metabolites in the soil, like lactic or acetic acid, that would hurt the roots of very young seedlings that you're transplanting. So you want to avoid having any phytotoxicity. We don't see phytotoxicity very often because, if you let these soils go for a week, other microbes in the soil will break down all these compounds and the soil will be fine. I have a collaborator at Penn State University who suggests checking soil fertility because you have added 6 to 9 tons per acre. It's going to add a lot of nitrogen to your soil, which is also very beneficial for most of our growers. And you just want to make sure that you're not adding too much fertilizer following use of anaerobic soil disinfestation. And then once that soil has reoxygenated, you can plant as usual. So with the ASD treatment, if your disease levels are very high, you might consider applying the treatment 2 years in a row. We've seen that works really well for our growers who have had long ongoing soil-borne disease issues. And a single treatment, while it would knock down diseases, probably you'll need to do 2 years of treatment in a row. And then after that, once you've gotten those diseases in check and you have some other disease management practices, you can consider doing the ASD treatment every 3 to 5 years. Okay, so that was a quick overview of anaerobic soil disinfestation and these soil-borne diseases. I really hope that you don't run into it, but unfortunately, as a plant pathologist, I know that you as growers will probably run into some of these issues in your own high tunnel or hoop house production. Now, feel free, we have time for discussion now, and I love discussing these issues. Feel free to email me at anna.testen@usda.gov, or my work phone number, and you're welcome to call it, is 330-641-2862. So.
A little fast.
All right. We have any questions?
Awesome. Thank you, Anna. So, yes, first question. Is ASD at all effective against Pythium?
Yeah, that is a great question. It is effective against Pythium. Pythium and Fusarium can be really, Phytophthora as well, can be very tricky to control with ASD though, because they're very fast growing pathogens. A lot of the other pathogens we're dealing with are slower growers. So we knock those down, and our other microbes in the soil can, benefit our plant and, help having healthier soil. But Pythium, you knock it down and there's still some that's surviving there. If the conditions are right and the soil is still moist and you have a good host, it's just going to bounce back. So you just have to be careful with that. Um, it will reduce, but it won't eliminate Pythium.
And I guess just to build on that, so say you go through something like ASD, you know you have high Pythium or like high levels of a soil pathogen, with then coming in with something like a trichoderma application, like,
Yeah.
kind of keep your approach stacked to try to
Yeah,
keep.
that's a great question. So combining ASD with other management tactics is great, and combining ASD with different biologicals is great. And we've actually found for trichoderma and bacillus, uh, so trichoderma and bacillus are two microbes that can survive ASD treatment. Bacillus is called a facultative anaerobe, so it can actually operate anaerobically. So you could actually apply your bacillus before treatment and
Hmm,
it would survive the ASD treatment. Or you apply immediately after the ASD treatment when you have a slightly different microbial community, and those biocontrols will survive a little bit better in the soil. So combining with biologicals is a very good option following ASD.
that's awesome. I have one other question about Pythium.
Yeah.
So I've heard that Pythium is a poor competitor, and if you
Yes.
have a healthy soil microbiome, that sort of helps, I guess, with tackling that Pythium.
Yes, that is correct. Pythium
Okay.
is a really poor competitor. That's a good reason why Pythium and hydroponic systems, I just shifted to hydroponic, but that's a good reason why in a hydroponic system, Pythium is such an issue because we don't have our full soil microbial community in these systems. And Pythium is just like, wow, there's a lot of moisture. I can reproduce, I can infect. But Pythium, um, a lot of Pythium control can also be managed culturally. So we want to make sure that we're not overwatering. Like you said, you want to have healthy microbial communities. Start adding some beneficial microbes early. Make sure that you have balanced fertility in your crop. Pythium also likes really fast growing roots, so don't over apply nitrogen. But it's hard. A lot of growers are starting with a peat or a different um, potting media, and Pythium can really thrive in those as well because those have reduced microbial populations as well. So
Hmm,
if you're having an issue with Pythium, look to an integrated approach and think about adding some biology back into your system as well.
for sure. Um, okay, so you talked about, wheat middlings or sort of these wheat products as a food source. Have you done any work with straw as a potential carbon source?
Um, I have not done work with straw. Straw you have to be very careful with because there's too much, the carbon to nitrogen ratio is too high. So if you did work with straw, you'd probably have to add something like a manure to bring up the nitrogen, because otherwise if you just added straw, like microbes would start to break that down and then they'd run out of nitrogen and wouldn't be able to break it down. So we
Okay.
want something that's really easy for microbes to break down. There are reports of people using straw. Hay, something higher in nitrogen would be better. We've had growers use what they call green chop, which is just cover crops that they chop up. And those usually have good carbon to nitrogen ratios. If you're using
Hmm.
a cover crop, ryegrass works well. You might consider using that as a supplement to something like wheat middlings, because sometimes it's hard to grow enough of that cover crop to to have that good efficacy, because you need 6 to 9 tons of acre
Yeah.
of biomass. That's quite a bit.
Yeah.
So you might need some supplementation.
What about coffee chaff?
Oh, great question.
Mhm.
That was studied in California and I believe worked. There's lots of waste products that you could use creatively. So, whatever you have locally, if you can get it up to that 6 to 9 tons per acre to like really inundate that soil with a lot of food sources, go for it. We've seen, yeah, coffee grounds, like tomato pomace. So the pomace is what's left over when you're making like juice or soup from something. Grape pomace, apple pomace, whatever you have available locally. Distillers dried grains. So if you have a brewery by you and can get some of those, even you can get fresh distillers grains, all of those have been shown to work pretty well.
Hmm. That's pretty cool. So yeah, I think if you guys have any questions about what you could use as a source, you can reach out to myself or you can reach out to Anna and we can kind of figure out a good plan of attack for you. I like the idea of cover crops, but
Yeah,
then realizing you also have to then grow the cover
you do,
crop
yeah.
before you do ASD, so it's a bit more
Yeah,
disruptive.
and the nice thing with cover crops is, I mean, if you were taking your tunnel out of rotation, you could grow the cover crop in there directly. But also, if you have a field nearby, you could grow a cover crop in that field nearby and then bring it in.
Yep.
So cover crops, I did some work on which cover crops would be effective, and we found that ryegrass, sorghum Sudan grass, and then your clovers were most effective for reducing
Hmm.
diseases. I think the clovers were more because those broke down really quickly because they also have a little extra nitrogen in them. But you could consider cover crop mixtures. It's just cover crops, you don't want them fully mature because then you have too high of a carbon to nitrogen ratio. So think about harvesting those a little earlier so that microbes can break them down easier, and then you do have to chop them up a bit. So there's a few extra steps with the cover crops, but it is less expensive than having to buy your own
Yeah,
cover crop.
interesting. Or you could potentially, if you grew your cover crop throughout the winter,
Correct.
maybe
Yeah.
incorporate that in the spring, little ASD.
Yeah, because I don't know, are any of you growers growing like a ryegrass cover crop in your high tunnels in the winter?
No, we're really, I mean, anyone feels free to speak up,
Yes,
but
it's
from what
like
I've
put in the
seen
chat.
anyways, we are just working on sort of integrating cover crops. We've done sort of two initial demonstrations of like, using row cover versus non-row cover and looking at seeding dates and looking at seeding mixes. So we're kind of just starting the, I would say, cover crop journey. We just did a great fact sheet if anyone's looking. Check out our Perennia website just to looks a little bit at the work that we did. We did a webinar with Judson Reed last March about cover cropping in protected spaces. And we're going to hope to sort of continue that narrative moving forward. So happy to chat with anyone about cover cropping in spaces as well. Okay, so a few more questions here about source. Have you seen alfalfa meal used as a
I
carbon?
have not. That is an option that we have available to us to buy at feed mills, but that should fit the bill in terms of, enough nutrients uh, and a decent carbon to nitrogen ratio that should break down really well. I know the alfalfa meal that we can get locally here is pelleted. Um, I think you can also get it like more as a powder type. Um, but just make sure that if you have pelleted that gets really well incorporated and evenly incorporated, because you just don't want like clumps in your soil.
Mm-hmm. And so you've sort of mentioned this carbon to nitrogen ratio a few times when talking about these sources. Do you have like an ideal range or like that
There's
perfect
been some
number?
studies looking at ideal ranges. I've just found like most of the time, this was a great question about straw. It's just like you know, think about like something that if you're composting and it wouldn't break down very quickly in the compost, like straw takes a while to break down in compost. That's not something you want to use in ASD. You also don't want to use a mature compost because most of the, easily broken down stuff has been broken down. So that's just one thing when I talk with growers is, just think about like if if you can compost it easily, it'll work really well in ASD. If it's something that would come out of the compost at the end, that's going to be, not have enough nutrients for the anaerobic soil disinfestation. And just make sure you can. I was clear earlier with that straw question. You can use straw. You just have to add a higher something else like manure, cow manure or poultry manure to add more nitrogen to that system so the microbes can break it down.
Awesome. And then so really like we're looking at not only if you have something that's not as easy to break down, we're not feeding those microbes the same way, so we're not getting that
Yeah,
same level of efficacy, but then we're also probably dealing then with a lot of residual material
correct.
after the
Yeah.
fact. Okay.
Yeah. So like example in Pennsylvania, Pennsylvania is a big mushroom producing state, and they've looked at spent mushroom compost, which is straw bread and horse manure. I think some other composts are added, and that was just too mature of a compost to be
Mhm.
effective.
Okay, awesome. We're gonna switch gears a little bit. I think that was a lot of our sort of source questions. So what soil temperature are you striving for, as opposed to air temperature?
Okay, great question. So soil temperature is above 68. Thanks for the conversion to
Yes.
20. And luckily the rest of my temperatures I do know in Celsius because I ran my experiments. So we found, really great temperature. Most of our high tunnel growers, when we monitor temperatures, that September to October can maintain around 25 Celsius. And that's average temperature, because daytime temperatures you can spike up really high. We've seen 35 to 40 sometimes on a sunny day when you have your sidewalls down. So 25 Celsius, if you can maintain that for 4 weeks, average temperature, you will be in very good shape. 30 Celsius, even better. Just when you're doing this treatment, drop your sidewalls, close your end walls, just keep those temperatures high inside your high tunnel, and that'll help insulate your soil as well.
And just, I don't know if you know these numbers off the top of your head, but if you're looking at some of the other sort of like steam
Mhm.
treatment or like solarization,
Yeah.
what sort of temperature ranges are we
Well,
looking at
I
there?
believe soil solarization, you need to get 35. I think it's 35 to 38 for a period of 3 to 4 weeks. So, and that's like consistent temperatures.
Yeah.
You could achieve that in a high tunnel in Ohio or Nova Scotia if you took it out of production in June, July. And then that's also, at least I know for us in Ohio, we don't usually have a constant stretch of sunny weather. If you have a couple cloudy days or a couple days of rain, that'll screw up the solarization. Steam sterilization, um, that I believe is you're right around boiling temperature when you put in there. And that's like a short 20 to 30 minute. You want to bring
Mhm.
that soil up to that temperature for 20 to 30 minutes. Now, if you're interested in soil steaming, um, myself and a collaborator just got funding to study that compared to anaerobic soil disinfestation. So we will have more information for growers in a couple of years.
Awesome. I'll be sure to bug you about it.
Yeah, well, and we're really interested in the impacts on soil health and our soil microbial communities because steaming and fumigation are really harsh on your soil microbial communities, whereas anaerobic soil disinfestation, you're kind of just shifting your microbial community and it'll return back to a healthy state during the cropping.
Okay, so yeah, we did have a question, what is the effect of it on the good soil biology? So it's
Yeah. So
not necessarily a clean slate.
yeah, I will talk a little more about that. So anaerobic soil disinfestation, at the end of the treatment, you have a lot of bacillus and clostridium. Now, people kind of freak out when I say clostridium because they think of it as human pathogens. There's a lot of beneficial clostridium as well. That's just in soil. So those are your anaerobic microbes. They're going to dominate. But once that soil comes back and is oxygenated and you add plants, your other beneficials and microbes that were there before, um, they will start to recover. And usually, when you compare the soil microbiome before treatment and then after treatment at the end of cropping, they're about the same. It's really nice, like consistent recovery. And then, somebody asked, why don't our pathogens recover? It's just the pathogens don't compete as well against what you have there.
Hmm,
So, yeah.
interesting. Um, okay, so let me see if we can answer some of these questions. Okay, how does ASD efficacy compare to steam for something like lettuce?
That's a good question. Depends on your pathogen, and then also it depends on your goal as a grower. So efficacy, both of them will reduce diseases. Again, pathogen dependent. Your goal as a grower, if you have 4 to 5 weeks to put aside for ASD, that will work. if you don't have 4 to 5 weeks, and you just want to get the treatment done in a day, steaming is your better option. So we just have to be, cognizant. Both will reduce diseases. I have seen more issues with steaming, with having stuff like Pythium coming back in, just because like you have knocked down your beneficials a little more dramatically than you do in anaerobic soil disinfestation. So just be aware of that. I do have a question too for growers. Like, is anybody in your group doing steaming? The major limitation for us in the US right now is just access to steamers. So, steaming used to be very common here in the early 1900s, when fuel was really cheap and you could just take a boiler into your into your field and steam your soil. But we've had a re-interest in this, but in the US, these steamers are $25,000 to $35,000. It's a very large investment.
Yeah. Yeah. So anyone feel free to speak up if you are using it. I don't think it's a common practice here, but feel free to correct me if I'm wrong.
Yeah, and a lot of our Amish farms, sometimes you can go and see some really old soil steamers, and some of those are being refurbished for use. So,
One other question in the chat here. So what is the actual mechanism of ASD that's killing the pathogens? Is
yeah,
it
it's a great question. So it's actually a combination of, you have the anaerobic nature of the soil. So lack of oxygen will kill some of our pathogens, because not everything can survive. And that's another reason why, unfortunately, Fusarium, Pythium, Phytophthora can survive and rebound after ASD, is some of those weird pathogens can actually go anaerobic and survive anaerobic conditions. And then you have your acids that are produced. So lactic and acetic acid are really good at killing pathogens. The metal ions that we have in our soil, we have manganese, iron. Those, when they're reduced, also will kill pathogens and have antifungal properties. The gases that are produced, sometimes we have like gases that include sulfur. Those have antifungal properties. And then the last thing are these different volatile organic compounds. So these microbes will spit out a bunch of weird little compounds, and those have antimicrobial activities. Now, it's a combination of all of these. Some pathogens are more sensitive to others, like something like a nematode, that's bigger and actually needs to respire. That's going to be more sensitive to a lack of oxygen. And actually, nematodes are the most sensitive to ASD. So if you have like a root knot problem, go ahead and do ASD. It'll knock it out even if soil temperatures are only 15 Celsius. So it's a combination, or this gamish of all these different changes that we have. Now, when this was originally developed as a technique, it was developed in Japan and the Netherlands. And like the original thought was, we know that flooding can reduce soil-borne diseases. We know that tarping to increase the temperature of soil can reduce diseases. And we know that adding some different soil amendments can reduce diseases. What happens if we do all three? And that's when you get the effective anaerobic soil disinfestation. It's a very complicated system, but that's why I've been working on it for almost 10 years.
Yeah.
Yeah.
No, that's awesome. It's a lot of moving parts for
Lots
sure.
of moving parts. But the actual application is not as complicated as you think. We've had a lot of growers adopt it, and it works well. And we've had growers also say that ASD improves the tilth of their soil. And we see some good benefits as well on soil health. So
I'm gonna put a
other
little pin in that comment
okay.
on the soil health.
Yeah,
How does it deal with weed seeds?
good question. ASD does decrease weed seeds. It depends on the size of the seed and the biology of the seed, how deep it is. But we've seen nice impacts in the high tunnels. If you have your control plot next to your ASD plot, there's a ton of weeds in your control plot and not many. Now, weed seeds that are a bit deeper might not have gotten hit. So, if you retill, you're going to draw those back up, but it does reduce the weed seed bank. So you will see a slight reduction in weeds. Again, that's very dependent on your species. There's some publications I can pull for you that show, what species of weeds are more affected.
Amazing.
Yeah.
What about root lesion nematode?
Yeah, nematodes are most sensitive to ASD. So yeah, ASD would knock out root lesion nematode. So
Awesome.
that's, and again, we found even at cooler soil temperatures, and this was not intentionally done, we just had a trial year where the temperatures got really cold. Even at average soil temperatures of 15 Celsius, we still saw a good reduction in nematodes. So
So yeah, if that's your main goal, then we're not necessarily aiming for that
yeah,
25, 30 degrees.
yeah, you'll get better efficacy, but if you are treating for nematodes and if for some reason your soil, the temperatures go cold, don't worry, you'll still get a reduction in nematodes.
Yeah,
Root lesion might not be as effective as root knot just because those are a little more free moving, but they still will be reduced.
awesome. And so you talked about a bit of a break, between sort of completing your ASD cycle and when you want to put those plants in the ground. Are there any problems like with prolonged breaks, like if you went for 4 to 5 months, waited through the winter?
Yeah, that's a great question. And just like the way that we've been doing it here in the Midwestern United States is treat in the fall, you could actually leave that soil covered until the spring and then just cut your planting holes, a week before you want to plant. And there's no harm in just leaving that soil cover that entire time and kind of letting it just simmer
Yeah,
for
awesome.
for those months.
Cool. Okay. So with that, I think we've addressed most of the questions in the chat. So I am going to circle back to our soil health or the soil health comment that you made. So like, have you looked at sort of changes in soil chemistry, pH? Like what's the
Yeah,
story
so
here?
it depends a lot on your soil. In terms of soil health, we do see a boost right after anaerobic soil disinfestation. You'll see an increase in active carbon. That's carbon that's associated with easily cyclable carbon by microbes. You'll see a boost in that. You'll see a boost in soil protein. You'll see a boost in microbial respiration. Now, so that means your soil is very different after it. Now, after cropping, a lot of those soil health factors, have returned to pre-planting levels. In terms of your nutrients, some growers see higher nitrogen levels following ASD. That's not something that I've seen too much. We've run our chemical panels, and there really hasn't been too much that's very different after cropping, but like right before you crop, you will have some residual nitrogen and improve microbial activity, and that'll help your crop for vigor as well.
Mhm.
pH, depending on your soil, because we have organic acids that are produced, some soils like sandy soils, you'll see a reduction in pH. I'm not as familiar with Nova Scotia soils, but in Ohio we have a lot of calcium in our soils, so we don't see much pH difference just because our soils are more buffered. Um, but again, those pH changes are different and or are sorry, are usually transient, and you don't have to account for that when you're planting.
Okay.
Mm.
And yeah, as we had kind of spoken about, like
Mhm.
we do see very salty soils as a result of
Yes.
sort of greenhouse production over time. We see that pH kind of go up as well. But there isn't, I guess it won't put you in the wrong direction. It won't make
No,
the
yeah,
problem
and
worse.
I have been asked about soil salinity and following ASD, and nope. I talked to a couple collaborators who work on ASD, and really none of us have a good answer. Um,
Hmm.
I'll have to ask my technician to check after our next experiment, the EC levels of our soils. But really, like, you're not, when you're adding your carbon source and you're adding your water, you're not adding too much extra salt to it.
Yeah,
But I am concerned about soil salinity as like a stress factor in these soil-borne diseases. So I'll be starting
absolutely.
research to look at that as well. So yeah.
And I think it's sort of as with anything, unfortunately, it's a very like multi-pronged approach.
Yeah.
So in a lot of the webinars we've been doing about soil health, we talk about taking the plastic off
Mhm.
maybe once every, 3 to 5 years when you're changing your plastic. So kind of work that into a rotation with ASD.
Yeah.
Obviously not at the same time, but
Yeah.
you know,
And that's a really good point. Our high tunnel growers here have started taking their plastic off. We are having a very rainy winter this year. It would be a great year if you had decided to take your plastic off to flush some of those salts, but our growers, we have a lot of Amish and conservative Mennonite growers who tend to very heavily fertilize, so they have some very high salt levels in their high tunnels.
All right. So if there are no more questions, which it looks like we kind of got through the big ones, I want to say,
There was one other question about drip irrigation.
oh my gosh, yes.
Oh no,
I'm glad you're paying
no,
attention.
there's a question about would there be increased benefits to having drip irrigation under the plastic to keep water level up? And that just depends. Like if you're treating flat, you could use drip irrigation under your flat plastic and treating just flat soil. That's easier because you can lay your plastic and then irrigate. It's less fun to like cover the edges of your soil when you're dealing with mud. It's a lot easier to work with dry soil. So I personally do like drip better. And then if you're treating in beds, definitely lay your drip, lay your plastic, and then irrigate your beds. I always do amend, irrigate, cover. You can do amend, cover, irrigate. It just depends on how you're treating. And if you're doing drip, just cover your soil, make sure your drip's down, turn it on, and then it's just a little bit easier to work with. And that if you watch the video in the virtual workshop, it explains that a little bit better. So with anaerobic soil disinfestation, there's certain things you're gonna have to tweak for your own high tunnel setting, but I really do prefer the drip tape rather than overhead irrigation for saturating our soil.
And just one final question to finish us off. And we're obviously talking about soil temperatures. So we're like having a soil temperature probe would maybe be a useful thing to have.
Yeah, if you've got one. Otherwise, I mean, I've in some of our early trials, we just had almost like a cooking thermometer that you stick in the soil. And it helps you as a grower. I'm sure you can get these thermometers really inexpensive. Just pop out into your high tunnel in the middle of the day on a sunny day and be like, oh, it's 30 Celsius right now. That seems good. And then check it first thing in the morning. You can also get probes that'll record for not that expensive.
Yeah.
So yeah, it would help you as a grower to make decisions when to pull the plastic.
Awesome. Right. Just really making sure it cooks.
Yeah.
Cool beans. All right. Well, I want to say thank you so much, Anna. That was awesome, super informative, great discussion. And not because you're a bad presenter, but like you said, lots to cover. Thank you everyone for joining us here today. Definitely keep an eye on the blog and registration for our next session in February. Yeah, if you have any questions, feel free to reach out to us and, happy to play where we can.
Thanks, everybody.
Have a good day.
Thank you. Thank you. Thank
Bye.
you.
Talia, I'll send you the PDF of the presentation.
Yeah, and I'll send that out to everyone.
Okay.
And
did you want the papers on weed seeds?
Yeah, that'd be awesome.
Okay, I think there's two like really good ones for the US that did like screened the common weed crops that they have in I think Ohio and Tennessee. So I will send those out.
Yeah, that's really cool.
Okay. I'll pull that right now so I don't forget.
Sounds good. All right. Bye guys.
All right. Happy New Year, everyone.
Yes.